University of Long Island. She went on to earn her PhD in plant pathology from University of California, Riverside in spring 2020. Under the advisement of Dr. Caroline Roper, she studied citrus tree associated microbiomes. As a graduate student, she was awarded the National Science Foundation Graduate Research Fellowship and the UC President's Dissertation Year Fellowship. Dr. Ginnan recently started as a postdoctoral researcher in Dr. Maggie Warner's lab at the University of Kansas. She's further exploring her interest in how microbes contribute to plant health by studying drought-induced adaptation in free-living and plant-associated microbial communities. We all welcome you, Dr. Ginnan. Thank you so much for agreeing to participate. And I think I'll hand over the mic to you now.
All right. Hello, and thank you for that introduction. Let me share my screen here. Hopefully this all works. Okay. So I did mention I'm in a hotel right now because I'm doing field sampling, and I'm just hoping that the hotel internet survives the talk. So if something cuts out, I'm just going to call right back. So just stay on the line. Let me get rid of this. Okay. Yes, so today I'm mainly going to be talking about my graduate research work in Caroline Roper's lab at UC Riverside. And then I'll just use one slide to explain my new project that I'll be working on in the Wagner lab at the University of Kansas. Okay, so we all know that there's microbial communities that are associated with eukaryotic hosts, and eukaryotic hosts that harbor microbes can be my cat, or it can be plants, and pretty much every surface has microbes on it. And when we talk about microbiome and the host together, I'll refer to it as the holobiont. And microbiota have been shown to provide extended traits and are linked to host health. And a few different ways that microbes can help hosts are by promoting growth, providing tolerance to abiotic stresses such as drought, and also preventing disease. When we talk about the larger picture, the phytobiome, we're thinking about everything that can affect plant health and also affect the microbial community associated with plants. And those factors include weather, geographical location or soil type, nutrient availability, pathogens and diseases and pests, also genotype and other host factors, and of course interactions with insects and animals. I'm really interested in studying all of these different components in it, how microbes change and how they affect plant health, because we can potentially use microbes as a way to manage some of these issues. So severe drought, pathogens, pests, and also growing plants in nutrient depleted soils. Today, I'm going to be focusing on citrus trees because that was what I worked on in my graduate research. And I'll be talking about both rhizosphere and phyllosphere microbes. I'll start with talking about how disease can induce plant dysbiosis and cause a microbial response. Then I'll move on to impacts of intermicrobiome competition and how that affects physical niche selection within the host. Then I'll talk about shifts in the microbiome that are associated with plant phenology or seasonal development stages. And lastly, I'll give a little update about my current project. Citrus is a very interesting plant. First of all, it's beautiful. but it's also very ancient. It was actually first described way back in 800 BC, and it's very recognized in flavor. Citrus flavors are used in everything from sodas to margaritas to obviously orange juice. And it's a very economically important and culturally important industry. In the US industry alone is worth $3.2 billion. These trees have very long lifespans. They can be in production for 40 years or more. And most of the time, these groves are generational groves. So it'll be passed down from a grandfather to a son to a grandson and remain in the family and support their lives for, for decades. Citrus trees are also evergreen trees, so they don't go through a dormant phase in the winter, which makes them extra interesting in terms of microbiology, because since it's active the whole year and they have leaves, then they also have a very active microbiome throughout the year. And right now, citrus trees are threatened by disease. And that disease has no cure. Whoops. Oh no, okay. And that disease has no cure, which I'll talk about in a little bit. And that's part of the reason that we were interested in looking at the microbiome, because there is no other way to combat the disease. So why not take a different route? So I'll just dive right into the disease section. And the disease that I focused on was Citrus Huanglongbing, also known as citrus greening, or shortened to HLB disease. HLB is a very devastating disease. It's drastically affected the Florida industry and cost them billions of dollars over the past 5 to 10 years. This disease is vectored by the Asian citrus psyllid, which was first found in 1998 in Florida. You can see that little psyllid here. And they feed on the plant, and eventually it causes this muddled chlorosis in the leaves, and you get shoot dieback and also root dieback. And the fruit becomes very bitter and hard and green, and it cannot be juiced. It cannot be eaten as a fresh fruit. So you're losing your production very quickly. And then the trees begin to decline. And eventually, within a few years, these trees are dead. And so these trees that are expected to produce fruit for these farmers for, 20, 30, 40 years, and within just a few years are all dead, that's very devastating. And a lot of growers cannot come back from that. And they have to switch to a different crop or, take other routes. So it's very devastating. This disease has been associated with citrus or Candidatus liberbacter asiaticus, a bacterium that is phloem limited. So it only survives in the inside of the plant. And it has currently not been cultured in pure culture. It has been cultured in a collective, a co-culture of other microbes in a biofilm. And so that's also one thing we thought, that it's surprising that sea lass can be called co-cultured, but not in pure culture, and that maybe it's very dependent on other microbes to survive. And sea lass was first found in 2005 in Florida. Since it's not culturable, Koch postulates has not been completed. So officially, it's called the associated bacterium with this disease. But for the purposes of this talk, I'll just call it the pathogen. And like I was saying, there are no effective controls or treatments currently. The only way that they maintain this disease is by controlling the insect vector. So this disease, it has devastated Florida, and it has moved over to Texas and started to cause problems there. And then it also is now found in California, which is currently the leading producer of citrus. And so they're very concerned that that will begin to affect California in the same way it affected Florida. But for our purposes, we're really interested in how sea lice may be impacting the native microbial communities. There is this model that I found that I think really describes different responses the microbiome could have to disturbances. And in this case, the disturbance is a pathogen. So a pathogen could come in and not change the microbiome, and the microbiome could remain healthy, and you would get a healthy phenotype, and we would consider this a resistant microbiome to this disturbance. Another option is that the pathogen could come in and disrupt the microbiome, and that microbiome could then recover and produce a healthy phenotype. And we would consider this a resilient microbiome. A third option is that this pathogen could come in, disrupt the microbiome. The microbiome could remain changed, but still produce a healthy phenotype. And we would consider this a functionally redundant microbiome. And lastly, and the case that is happening currently, is that a pathogen will disrupt the microbiome, it will remain changed, and it will produce a bad phenotype, an unhealthy tree. And we consider this dysbiosis. So what we're hoping is that by understanding how this pathogen is impacting the microbiome and how these changes are causing a dysbiosis, that we might be able to rebuild a microbiome or engineer a microbiome that could be resistant, resilient, or functionally redundant. So our first step is that we went to Florida, which is currently under very high disease pressure. It's estimated that between 95 and 100% of field trees in Florida are infected with this pathogen. So we sampled a few different orchards that had various varieties and age of trees. And we sampled the roots, the leaves, and the stems. We were also interested in this survivor tree phenotype. So we developed a disease rating scale from 1 to 5, 5 being nearly dead, and 1 being appearing healthy but still infected. What we were told by different growers and also by this other researcher in Florida is that some trees, even though they're under high disease pressure, appear to be healthy in a grove. So because we know that citrus trees are clonally produced, and so a whole grove is genetically identical trees. But if some trees are doing better than others, we thought maybe that's because they have a particular microbiome that is more resilient to the disease. So in 2016, we went into a grove and we saw that there were some trees that appeared to be healthy, even though they were surrounded by unhealthy trees. So we sampled those survivor trees and their non-survivor tree counterparts. And we rated everything on a disease scale of 1 to 5. We came back in 2017 and we sampled those same trees, both the survivors and non-survivors, and measured their disease ratings. And we did the same thing in 2018. And what we found is that these trees that we originally marked as survivor trees, well, they did increase in disease rating, they still appeared to have a decelerated disease progression. So they weren't remaining healthy, but we did redefine survivor tree as being decelerated disease progression trees. And we made a metric to bin those trees. So then we developed our 2 study questions from the knowledge we gained from the field. We were interested in understanding what microbial changes are occurring across disease severity. And then we were also interested in seeing what microbial differences might be affecting decline. So what microbiome, how is the microbiome different in survivor trees versus non-survivor trees? In order to look at the microbiome, we used amplicon sequencing for bacteria and fungi. And we also did some qPCR work to look specifically at citrus pathogens, that being Phytophthora and some citrus viral pathogens. I'm not going to go into any virus information today, and I'll mostly just be focusing on bacteria and fungi. The first thing is when we looked at our data, at the largest scope, we saw that tissue type was a major driving factor of community composition. So you can see here, this is a beta diversity plot, a PICO-A. And I don't know if you're familiar with these plots, but each point represents an entire microbial community from one sample. And if the points are close together, those communities are more similar to each other, and if they're farther apart, they're less similar from each other. And what you can see here in our bacterial and our fungal communities look pretty much the same, and that roots, leaves, and stems are all significantly different from each other. So moving forward, we just analyzed the tissue types separately. Okay, so then we started to move into our first question. What changes are occurring across disease severity as the disease severity increases? And again, here is a beta diversity plot, but this time we've separated the tissue types, but I'm just going to be focusing on one above ground tissue type, the leaves, and one below ground tissue type, the roots, feeder roots. And here I have the points colored by their disease rating. And from the statistics, we know that disease severity is a driving factor in community composition, but these plots are pretty busy. So what I really want you to focus on is that our healthiest trees, our disease rating 1 trees, their community composition is very different from our disease rating 5 or our sickest trees. And so right away, we know that there are drastic changes in the microbiome, and that potentially we can track that across the disease ratings. But we wanted to know what's happening at a taxonomic level. So the first thing we looked at, of course, was the relative abundance of the pathogen associated with this disease. And here you can see across the disease ratings that the relative abundance of the pathogen does increase as the disease severity increases. Except for DR5, we thought this was very interesting that there is a drop in relative abundance, and we think that this is because at this point, the tree is so unhealthy that the environment cannot even support the pathogen. It could also be, since this is relative abundance, that there is a large influx of secondary pathogens or infections, and that is causing this drop in relative abundance of the pathogen. And so we wanted to look at things that are changing very early on in the disease, our DR1, 2, and 3. And we did notice that there is some microbes that have a significant drop in relative abundance very early in the disease. And interestingly, we also found that there were some microbes that had this pattern where they had an increase and then decrease. And this really alerted me because it kind of looks like a response to the disease. And when we went to the literature, we were surprised to see that that some of these microbes that had this response-like pattern were known plant growth promoters and can produce antimicrobial compounds, and that some of them are also biological control agents in other systems. So it kind of makes sense that they are potentially responding to this dysbiosis. And of course, the loss of these beneficial microbes could contribute to some of the symptoms we see in the tree. Another thing that we looked at is Phytophthora species. So Phytophthora is a root pathogen that is a very significant problem for citrus. And what we saw is that very early on in the disease, there was a 34% increase in incidence of Phytophthora from DR1 to DR2. So as a farmer, if 34% more of my trees are getting infected with a root pathogen, that's a very biologically significant result. Later on in the disease, we saw that there were significant increases in a lot of fungi, and some of them known to be citrus pathogens. And then also we saw an increase in saprophytic microbes. And so these increases in other pathogens could contribute to the disease symptoms we see. And the saprophytes are probably coming in late in the disease to feed on the dead and dying tissue of the tree. Other patterns we saw in late disease was a significant decrease of beneficial plant growth promoters, and also an increase later in disease of antimicrobial producing microbes. This was also, again, surprising to us. And we think that it may be kind of a last line of defense, or again, another response to the disease. Then we wanted to look into our second question: what microbial differences are there between our survivor trees and our non-survivor trees? So we focused first on our survivor trees, which you can see here, and there were some microbes that had significantly higher relative abundance in our survivor trees than our non-survivor trees. And again, when you went into the literature, we found that these are known to be plant growth promoting microbes and produce antimicrobial compounds, and are also biological controls in other systems. On the other side of this, there were some bacteria and fungi that were more abundant in non-survivor trees versus survivor trees. And these just happen to be citrus pathogens and also produce starch degrading enzymes. So another symptom I forgot to mention is that the phloem of the plant actually gets clogged and there is a starch buildup in the leaves. And so we think that perhaps having an increase in a microbe that produces starch degrading enzymes may actually be a way to combat this disease. And we found some microbes that are helpful for getting nutrient to the plant. And we thought, again, that this might be some kind of last line of defense. The sickest of trees are recruiting microbes that may be able to help combat some of the symptoms of this disease. So overall, we developed a model of what we think that we speculate could be happening. First, early on in the disease, there is a quick decline of potentially keystone species and also beneficial symbionts. And this causes a dysbiosis. And then early on in the disease, there are some beneficial responses that you see here of microbes that are known to be plant growth promoting. Then later on in the disease, you have an increase in other pathogens and good competitors, and you have a second beneficial microbial enrichment, which is mainly focused on microbes that are producing antibiotics. And then finally, late in the disease, you have an increase in saprophytes that are surviving on the dead tissue. And it just so happens that our survivor trees had a lot of these early microbe responses patterns, and the microbes in our non-survivor trees had a lot of these late response microbes. And so what we were thinking is that potentially if we could maintain these beneficial microbes and also try to work on combating these secondary pathogens, that we might be able to sustain these trees for longer in the field without actually directly trying to get rid of CLAS. So moving on to the second story in my talk, I'm going to be looking at the impacts of intermicrobiome competition on colonization. The first thing we had to do when we wanted to look at specific interactions of microbes in a host was to develop an Axenic citrus system. So germ-free mice have been used for decades to help understand what's happening in the microbiome. And there have been other axenically grown plants, but their systems cannot support a large citrus seed. And also, it's sometimes very difficult to add microbes back to these agar systems. So I tried lots of different things in my PhD to grow germ-free citrus plants. I grew them in test tubes. I put them in media, but eventually I found the flow pot system that was developed at Michigan State, and I basically modified it so that it could work for citrus. And so what you use are these glass beads for drainage, a soil-like matrix that can be fully sterilized. And this way you can water from the bottom by just injecting your microbial solutions and evenly water. And I also found that you need to actually peel the seeds bare and then bleach them and to get them sterilized. And then put them in these boxes that have a HEPA-like filter on top to allow for gas exchange, but to keep everything in the box sterile. And so we could grow plants for over a month in this system. So after we developed this system, what I was really interested, going back to my first story, is how leaves and roots have very different microbial communities. And when I was looking in the literature, what it's thought is that the soil acts as a reservoir of microbes that then enter through the roots and go through some ecological filtering to the phyllosphere. So I was very interested in how this happens, and is this controlled by the physical and chemical components of the plant being different in the leaves and being different in the roots? Or is there more do the microbes have more control over that? And is it actually more competition-based niche selection? So what I did is I used my Axenic system and I added a mixture of soil microbes to a seed. And as the plant grew, I let the microbiome self-organize. So naturally, what wanted to be a phyllosphere microbe, it did that. And what wanted to be a rhizosphere microbe, we let it do that. And then what I did is I cultured from the surface of the leaf, or the episphere, and the endosphere, or inside of the leaf, and did the same for the roots, the outside and the inside of the root. And I ended up finding, just randomly selecting bacteria that were primarily only isolated from that single compartment and not from the rest. And it just happened that these were what I identified them as. Then with my 4 isolates, I added them back to plants at a seed stage. So I inoculated a seed with just a single strain, And then I did a mixture of all 4 of my isolates and inoculated those back. And I also did some pairwise inoculations, but I'm not going to talk about those today. And then I did the same way. I isolated from each of those 4 compartments, the inside of the leaf, the outside of the leaf, the inside of the root, and the outside of the root. And you can see here my isolate, shortened isolate names and the compartments. So this was when this isolate was inoculated to the plant alone, it could colonize all 4 compartments, which is easily seen because this microbe is yellow. It's pigmented. And here you can see, well, my negative control was sterile, and that when I inoculated all 4 isolates together, that Chronobacter's colonization was drastically reduced and actually eliminated from one of the compartments. And another way to look at this that will be easier than trying to point out all the differences is with this. So here I have my solo inoculations and my group inoculations. And if it was always re-isolated out from that compartment, it's green. And if it was not re-isolated out of that compartment, it's gray. And if it was sometimes re-isolated from that compartment, it's yellow. And what you can see across the board is there's a lot of green here and a lot less here. So basically, in a group inoculation, hold on. I'm still in here. Okay. The housekeeping for the hotel, sorry. So what you can see is that the colonization is reduced when there are other microbes present. So this alerted us that, yes, there is some interactions that are controlling the colonization within the microbiome. So of course, what we wanted to do is then see who's inhibiting who. And we did a ton of different interaction assays, looking in liquid and doing all different types of streaking. And basically what we found is that in vitro, there were no interactions between these microbes. And so we were really confused. We were trying to figure out what's happening inside the plant that's not happening in vitro. So we decided to do some whole genome sequencing and some quality filtering, and we assembled those genomes and annotated them. And then we identified antibiotic resistant genes using the CARD database, and we used anti-SMASH database to predict secondary metabolites, antibiotics and siderophores and all of that. And then I also did some metabolic modeling using Model Seed, and I did gap filling with different media inputs. So I predicted what would happen if this microbe was grown on LB, which is the media type we used in our in vitro assays, and then also predicted what metabolites are produced when this microbe is grown in complete media. So basically its full capabilities, which we thought would at least tell us what might be happening in the plant. Because there's no nice plant media that you can input into these models, since everything is so variable within plants. So we just used complete media. And then we compared those outputs. And what we found is that here are our isolates. They did have varying resistant genes, varying number of resistant genes and secondary metabolite genes. And some of them did produce antibiotics. And they also, 3 of them had different siderophores, predicted siderophores based off their genetic data. And from my metabolic modeling, what I found is that 2 of these microbes could produce metabolites in complete media that they could not produce in LB. So I will say that this is a bacillus strain that we had some trouble growing, and we couldn't re-isolate it all the time. So for these predictions, we actually removed it and just focused on what could be happening with these three, since they were always able to be grown and isolated. And one of the main things we found, obviously, is that vitamin K2 can be produced in complete media and potentially in the plant, but not be produced in LB. But what does that mean? Well, we made this model from our genetic predictions. So here, this Burkholderia strain, based off from our predictions, can produce beta-lactam antibiotics. And these two other strains, they actually are predicted to produce bacterial lipocalins, which are a resistant protein that can bind this antibiotic and block it from getting to those bacteria. And so we think that in vitro, this is what's happening, is that these bacteria are able to block this antibiotic. In planta, the same thing is happening, that this Burkholderia strain is producing antibiotics, and that this Klebsiella and Chronobacter are producing bacterial lipocalins. But this Burkholderia, we showed, could potentially be producing vitamin K2. And what I learned based on literature is that vitamin K2 can bind bacterial lipocalins with higher affinity than this antibiotic. So what could be happening is that it's producing the antibiotic in vitamin K2. The vitamin K2 is blocking these resistant genes, and now the antibiotic can get through and inhibit these other isolates. So I'm hoping that the next graduate student following after me will follow up on this and prove my prediction. One other thing we were thinking about in the leaves is that in the leaf, there is more competition for iron, and the different types of siderophores had different affinities for binding iron. And so it seemed like the Burkholderia and Chronobacter strain could outcompete the Klebsiella strain because they have high iron binding affinity siderophores and also rapidly secreted siderophores, whereas Klebsiella kind of had a weak, wimpy siderophore. All right, so just getting to my last part, I'm gonna have to speed up a bit, so sorry about that. Basically, plant phenology is the timing of annual reproductive and developmental events. And so basically flowering, fruit set, all of that. And phenology can affect the yield of the plant, the adaptability to climate change, the pest efficacy, pest treatment efficacy, growing distribution. It can affect potentially pollinator populations. There's some theories about pollinator and plant phenology mismatch. And it could also affect human allergy seasons by causing early allergy seasons or prolonged allergy seasons. And of course, we are facing changing climates and more and more pests and pathogens. And so understanding and defining phenology microbiome interactions could help us to stabilize and manage plant phenology, even in the face of these issues that we have. And of course, there's always the struggle between trade-offs of putting energy into defense or reproduction. So by understanding the phenology and microbiome interactions, we're hoping that we can help to overcome some of those issues. And it's also been shown recently that rhizosphere microorganisms may be able to influence phenology, specifically flowering times. And it has been shown also that microbiomes are impacted by developmental stage, but most of this research has been done in annual or short-lived perennial plants that have a short growing season. And so there is this issue of tissue age versus developmental stage and how you can't really separate those variables in a lot of plant species. So if you have, this rice here and you sample this leaf and you sample this leaf, chances are they have a different microbiome, but you can't really tell if the differences in the microbiome are because this plant is flowering, or is it just because this plant is more mature and older and has had time to recruit more microbes. And this is part of the reason, the benefit of working with citrus. So citrus trees are evergreen trees that are active throughout the year. And of course, their microbiome is also active throughout the year. And the trees that I sampled were actually 20 years old. So they have had a long time to collect a microbiome and maintain it. And I could sample the same aged leaves and roots throughout the entire year. And I looked at the phyllosphere and rhizosphere in my study. So I sampled across 7 different phenological stages of citrus: mature fruit, flush, flower initiation, full flowering, fruit set, fruit development, and color breaking. And it's good to note that citrus phenology is very well documented, and it's because citrus fruiting is an important industry. And people are actually using models to predict when citrus trees flower. That way they can spray pest treatments at the most effective time, and that way they can produce as many fruit as possible. But one thing that all of these models fail to do is incorporate microbial activity. And of course, if we understand how the microbiome is changing across these phenological stages, and then we're getting closer to understanding how they affect or maybe co-regulating phenology, and that could help us develop better models in predicting when flowering will be. So across all of these phenological stages, I just took my samples and looked at the number of OTUs, or the alpha diversity. And here I have my bacteria and my fungal communities and leaves and roots down here. And what you can see is that there are changes in alpha diversity across the phenological stages. And here I just shortened the phenological stages to, flush is F and flower initiation is FI, and so on. Hello?
Go ahead, Nichole.
Okay. Sorry, I know I'm going to finish up here soon. And what you can really take away from this is that fruit set has the highest alpha diversity and in the leaves, and in the roots, well, in bacteria root community, the highest alpha diversity is in flowering, and that the root community has less changes, less variable, and more stable across the year, whereas the leaf community seems to be more variable throughout the year. And when we look at our beta diversity stats, we can see that across the board, phenology is the largest driving, or is the major driving factor of community composition. And that, well, significant, many of these other factors that we measured, such as fertilizer and rainfall and temperature, have a very small amount of impact on community composition, as you can see by the R squared. And so phenological stage is a major driving factor of community composition. And as you can see here, phenology explaining 27 and 22% of the variation in the data in the leaf communities versus 9 and 12% of the variation in the root communities. You can see that phenology is having more impact on the leaf communities than the root communities. So what I decided to do is take a core microbiome from each individual stage. So what is the core microbiome of mature fruit? What is the core microbiome of flesh? And so on. And then if a genus was core to 0 stages, I removed it from my analysis. And if it was core to 1 to 5 stages, I considered it to be a temporal core or a stage-specific core. And if it was core to 6 to 7 stages, I considered it to be a permanent core microbe because it is in high relative abundance throughout the year. And what I found is, you can look at the percentage here, that leaf bacteria had the lowest percentage of permanent core, and it actually had the highest percentage of temporal core. And since we were really interested in finding genera that correlated with specific stages, I focused a lot on leaf bacteria, which I will talk about now. So what I did is all of our temporal and permanent core genera from our leaf bacterial compartments, I made a phylogenetic tree of those. And basically here what you can see is, again, the phenological stages: flush, flower initiation, full flowering. And if there is a box under that category next to a genus, then this was core to flush, and this was not core during flush, and so on. And so these ones that are across the board have these colored blocks, they are our permanent core. And if they just have a few blocks, then they are the temporal or stage-specific core. And what I did is, by forming this plot, I was able to look at patterns, phylogenetic patterns of abundance. And so basically, I found that gamma proteobacteria are consistently abundant during full flowering, and that most beta proteobacteria are core to 4 or more stages, which might suggest a more generalized function in the system. And then our strongest pattern I saw here is that actinobacteria are associated with fruit set and fruit development, and are never associated with mature fruiting, flower initiation, or flowering, except for this one genus, Bifidobacterium, which I'll talk about on the next slide. And so this does show evidence of phylogenetically inherited traits and possible function, and also possibly functionally redundant roles within clades. And so I zoomed in and looked at some of these genuses and their specific relative abundance across these stages. And what I found is that some of them had a gradual enrichment to flowering, whereas some of the other ones, they had a sudden enrichment during flowering. And what we kind of started to think was that this appears to be, we speculate that this is a pattern of natural selection, and that we speculate that this is a pattern of dispersal or introduction of a new organism into the system. And when we looked in the literature, we did have some support for our hypotheses on that. So this genus has been associated with low ABA and high GA, and is a known phosphate solubilizer. And we know that phosphorus and GA promote flowering, and that ABA delays flowering. So making these connections strengthens our hypothesis that we hope to investigate further. And when we looked into the literature, we were excited to find that these microbes, these genera, are actually associated with bees. And so you can imagine that a bee will visit a plant during flowering and could disperse or transmit microbes to the plant. And what I talked about before is there is that one genus that didn't follow the phylogenetic trend of actinobacteria, and come to find out that that was the one that we predicted to be an immigrant into the system. And so it makes sense that if this microbe did not develop in this system, that it would have a different colonization pattern, and also here that it was dispersed during flowering. Then when we looked at some other, okay, I'm running out of time, so I'm just gonna say, we looked at other correlations and enrichments during different stages, different phenological stages, and we did find microbes that are significantly enriched during fruit set and development, and that they had a ton of different ones that were during that time, and that those did have literature to support the fact that they may be important for healthy fruit development. And sort of the model that we developed here is, when we zoom in on these specific stages, is that there are microbes that have a gradual enrichment from flower initiation to full flowering, and that during flowering, the plant is probably more exposed to interactions with insects, and that those insects may be dispersing stage-specific microbes to the plant community, and those may be incorporated and have a function in the system. Then we have fruit set and fruit development, which brings in a whole new range of microbes that are enriched during that time and could be also co-regulating some of those phenological stages. Then I'll just briefly, I have one slide on what I'm working on with Maggie. Yes, so I'm investigating microbial adaptations to drought, and I'm interested in looking at both free-living microbes and host-associated microbes. And we have a domesticated maize and a non-domesticated relative, and we think that drought adaptations may be, the community may evolve differently if there's a host present or if there's not a host present. And so we're sampling across a natural drought gradient, which is the state of Kansas. And we're using metagenomics and metatranscriptomics to look at community level shifts and adaptations. And then we're also doing some simulated drought experiments to then look at more population level mutations that could be making communities more resistant to drought. And so I've only been a postdoc in Maggie Wagner's lab for a few months now, so we're just really getting this project together. And since I'm almost out of time, I'll just say all the conclusions. And I just really want to thank my graduate advisor and my graduate lab at UC Riverside, and of course my new lab at the University of Kansas, and all the collaborators that made the projects I showed possible, and the funding agencies. And I'm gonna stop sharing. Okay.
Thank you so much, Dr. Ginnan. We appreciate your time, and I think it was a very interesting talk. And one of the things that I really liked was the Axenic citrus system, the germ-free plant. You have to optimize. I'm sure you must have optimized so many different conditions to get to a point. So I think we can, we only have 5 minutes available for questions and answers. So is there any question for Dr. Ginnan? I see Mohammed's hand. So Mohammed, you can please go ahead.
Yeah, so thank you for this very interesting presentation. I have an experience in citrus farming, and as I know, the roots are wild type, came from seeds, while the upper part is a variety joined by grafting. So almost of the citrus trees are developed using this technique, at least in the Middle East, I don't know in the East. So as you mentioned, the roots and leaves microbial communities are different. Do you think this may affect this difference?
Oh yes, it definitely does. But it's been shown across all different plant systems that whether they're grafted or not, The leaf and the root communities are significantly different from each other. With citrus and some other crops, there is that added feature that there is a rootstock that's genetically different from its scion on top. And there's a research team I know that is looking at how if you have all the same scion, but it's connected to a bunch of different rootstocks, how that changes the microbial community. But I don't think they're very far into that research. So maybe in about 2 or 3 years, we'll see a publication on that, and it would be very interesting. And of course, that could affect disease, because there is some rootstock varieties and scion varieties that are a little bit more tolerant to HLB. But there is no resistant varieties, marketable resistant varieties at this time.
Okay, thank you.
Thank you so much. We have one of our professors, Dr. Suhaj Baji, asking you a question. And she asks, in our study, we demonstrated that starch buildup in leaves is associated with the thylakoids and grana of leaf chloroplasts. These starch granules were directly related to increase the resilience of the plants to drought. Do you think your observations of starch granules in leaves makes tree are resistant to abiotic stress?
It definitely could be. And that is a good point, because what's happening inside of the plant is that the phloem, which is the main transport system within the plant, is getting clogged up as a host response. It's clogging up to try to block the pathogen off. And that is basically causing almost abiotic stress in the plant. Because if it's clogging off its main source of energy, then you are going to see some nutrient stress and also probably some water stress. And so maybe that starch buildup is the host's way of reacting not to the pathogen, but to the host-induced stress that is resembling abiotic stress. It could be. It's a very complex disease. Yeah, I like that thought a lot. Thank you. Something to think about.
So I think it's almost okay. I see another hand, Meha. So maybe last question from Meha.
Thank you so much for your talk. Actually, one of your slides, you mentioned that the rhizosphere is affecting the flowering time. So have you observed any one particular bacteria which is in the roots and in the leaves, and which is affecting the flowering time in citrus?
Okay, so I did not. So I'm doing correlations, not necessarily causations. I did start doing some causal and functional assays where I had isolated some of those microbes that I predicted could potentially be affecting phenology. But because of COVID, I did not get to finish those projects before I had to graduate. So I'm hoping that someone will follow up. And we have strong speculation of a couple different microbes having a significant role in flowering time in citrus. But I can't really, I can't give a good answer right now because I haven't done those experiments.
Yeah, it's really interesting to see the flowering time genes in the roots. So no, thank you.
Yeah.
Okay, I think we will wrap up now. So thank you so much, Dr. Ginnan. We appreciate your time, and we really enjoyed the presentation. And thank you so much again.
Great. Yeah, thank you so much for your invitation. And I know I didn't give a lot of time for questions, so please email me with any questions you have about my projects, or if you just want to chat or anything, feel free to email me at any time.
Thank you so much.
Okay. Goodbye. Thanks again.